let's look at aerobic cellular respiration. Now, aerobic cellular respiration, at least from our point of view, is going to be three separate but connected metabolic pathways for essentially taking that really, really stable glucose molecule and turning it into ATP energy. Those ATP energy molecules can then be used inside of our cell to do things like run those pump proteins. Remember when we talked about the pump proteins doing active transport, using ATP energy in order to move ions or move great big molecules like even glucose back and forth across a plasma membrane. ATP energy molecules can be used by the motor protein. Remember that video when we saw the motor protein with the great big organelle above, I want to call it its protein head, but we know that's not actually true. So it's got that great big organelle and she's walking along, and every time it takes a step, that motor protein is using a molecule of ATP. And so inside of our cell, we have so many things that are using ATP energy molecules to get things done. So aerobic cellular respiration is one of those metabolic pathways that's going to allow us to use glucose and turn it into ATP energy and get lots of things done. And I know it's complicated. I know there's a lot of steps and a lot of enzymes involved, but really, this is the best way to get energy back out of glucose. Now remember, glucose is a monosaccharide. And when monosaccharides bond together, they make a polysaccharide. And one polysaccharide that we studied was cellulose. And cellulose is just lots of little monosaccharides all linked together. Cellulose has a linear straight shape. It's super, super stable. It's so stable that when we looked at the redwood tree, we could see how all of those glucose molecules bonded together in cellulose can stay stable for thousands of years and can hold that tree upright against gravity, against all of the weather, against all of the things, including people carving tunnels out underneath the trunk of that tree so that they could drive their cars through. Like, that's how amazing cellulose is. That's how amazing monosaccharides like glucose are. There's a lot of energy bound up in the carbons, hydrogens, and oxygens of that glucose molecule. So we're going to need this specialized system. We're going to need all of these enzymes to get the electrons back out, to get the energy back out of this stable glucose molecule. So remember our formula for aerobic cellular respiration. Our input is glucose and oxygen. Through the process of getting energy back out of glucose, we're going to make carbon dioxide and water. But most importantly, we're going to make between 30 to 40 ATP energy molecules. The reason why I've got a range there is because I'm familiar with biological systems. There's always going to be some variation. Different mitochondria will be more efficient. Different mitochondria in different species. Say a yeast versus a human versus a cat versus a dog. All of our mitochondria work a little bit different. So sometimes they may make 36. Sometimes they'll make 37. Sometimes they may only make 33. It all depends on a lot of different variables. So instead of just saying one glucose molecule will make 36 ATP every single time, I want to account for the idea that biological systems have variance, and there's always going to be a range. So we'll get between 30 to 40 ATP energy molecules. But that's a lot of ATP energy. That's 30 to 40 steps for our motor protein from just one little glucose molecule. So in order to talk about these three separate but connected metabolic pathways, I actually wanted to focus in on a plant cell. Now, plants, remember, a lot of them are autotrophs. They're self-feeders. So in my plant cell, I've got my cell wall, and I'm making it look very geometric here, very straight. The secondary line is my cell membrane, and this should be pressed up tightly. There shouldn't actually be any space. I wanted to be able to show you those two structures, the cell wall and the cell membrane. So then everything inside of here, this is my water balloon, everything inside of here is going to be the cell organelles, the cytoplasm. This is where all of those metabolic pathways are going to be happening. Now I squished over here down in the corner the nucleus that has the DNA, our rough endoplasmic reticulum for folding proteins, our smooth endoplasmic reticulum for making lipids, our Golgi apparatus for several separate sacks of enzymes that are going to be doing different types of protein folding, and our chloroplast organelle. Our chloroplast organelle, as we know, is doing the light and the dark reactions. The light reactions are happening in thylakoid membrane to make the ATP and NADPH energy that will be used by the Calvin cycle, or the dark reactions in the stroma, where our rubisco protein is going to be using that ATP and NADPH energy to force carbon dioxide to become glucose. So all of that action, hopefully you'd made those drawings and looked at those pictures in the lecture slides in the textbook. All of that is happening over here in this little small oval. The reason why I wanted to shrink everything over here is that I wanted to blow up and really focus on aerobic cellular respiration. I wanted to focus on the fact that our plants not only do photosynthesis to make glucose, but then they also can do aerobic cellular respiration. This is why they're self-feeders. I wish I could do this, then I wouldn't have to think about what I need to make for dinner. All I've got is the mitochondrion and the enzymes to do glycolysis, so I still have to go out and find my food. I just don't get to sit there in the sun and make my own food. So, our plant cell is going to be doing aerobic cellular respiration, and the first step is always going to be glycolysis. Remember, glyco, or that prefix at beginning, glyco refers to glucose, and the ending lysis means breaking. So the first step is we have to grab that stable glucose, and we have to break it in half. This is going to be the energy investment phase. Now, I'm not getting into the details of the energy investment phase, but note that our stable glucose molecule is going to require the enzymes in glycolysis to use some ATP energy. So I'm going to draw a little glucose molecule, and I'm going to split it in half. This is going to require ATP energy. Our enzymes are going to take that broken glucose and eventually turn it into 2 molecules of pyruvate. So once the enzymes have broken glucose, they start to strip away hydrogen. They start to strip away electrons. So part of what we will get through this process of manipulating glucose into pyruvate will be NADH energy, and we'll get some ATP energy molecules. We'll get a total of 4. But because we needed 2 ATP in order to break the glucose in the first place, only 2 of those ATP energy molecules can be used elsewhere in the cell by something like the motor protein or a pump protein. So all of this work of glycolysis is happening in the cytoplasm. We've got lots of enzymes that are involved in breaking glucose and turning it into pyruvate, stripping away those hydrogens, stripping away those electrons, oxidizing or beginning to oxidize our glucose molecule. So lots and lots of different types of cells can do this. You don't need a specialized organelle to do glycolysis. Glycolysis is a very old metabolic pathway. It may have been one of the first ways that cells were trying to work out how to use and transform energy. So we've got the glucose molecule broken. We've got it transformed into pyruvate, but pyruvate still has lots and lots of energy. So this is where our mitochondrion organelle becomes so incredibly important. Now, our mitochondrion organelle has 2 separate membranes. It has this outer membrane. I'm just going to title it outer membrane, outer M. It also has this inner membrane. And I'm just going to draw this inner membrane as kind of a little squiggle. I know that it's actually really, really deeply folded, and there's not much space between the inner and the outer membrane. Now, our inner membrane gets a special name. It gets called the cristae. And the reason why it's getting that special name is because of all of those intricate folds. In other words, our inner membrane has lots and lots of surface area. In between the outer and inner membrane is the intermembrane space. So I'm going to title it inter-M space. And then deep inside of our mitochondrion, we have a biochemical goo called the matrix. So our broken pyruvate will go straight into our mitochondrial matrix. Now, I don't want to cover the details of the prep reaction. If you're going further in biology, you'll learn how the pyruvate gets grabbed, and there's a little bit more that has to happen before that pyruvate can go directly into the citric acid cycle or the Krebs cycle, depending on how you've learned about it. So I'm just going to draw our pyruvate going straight into that mitochondrial matrix, going straight into our citric acid cycle. Now what's going to happen with our citric acid cycle or our Krebs cycle is enzymes are going to grab a hold of pyruvate. They're going to strip away hydrogen. They're going to strip away electrons. They're going to continue the oxidation of our original glucose molecule. In the process of doing this, they're going to make NADH energy and FADH2 energy. They'll also make some ATP, but really the focus and the emphasis here is on the NAD and FAD energy molecules. We'll talk about those in just a minute. We'll only get a few ATP energy molecules. On the process of stripping away hydrogen, stripping away electrons, our enzymes are also going to produce some carbon dioxide. This carbon dioxide is a gas. Really high concentration here in the matrix, low concentration in the cytoplasm, low concentration in the atmosphere. Our carbon dioxide is going to diffuse out of our mitochondrion organelle. So, glycolysis broke the glucose molecule. Enzymes got it transformed into pyruvate. That pyruvate went right into our mitochondrial matrix, right into our citric acid cycle. Whole point of the citric acid cycle, all of those enzymes continuing to manipulate that broken glucose, continuing to strip away hydrogen and hydrogen and electrons from the pyruvate, oxidizing the pyruvate, is to make NAD and FAD energy. So the whole point of the NAD and FAD energy is for the electron transport chain. So I'm going to bring this inner membrane just a little bit bubbled out, because what I want to draw in my inner membrane, my cristae, is I want to draw this sequence of proteins, the electron transport chain. They're all embedded here in the membrane. So we have our electron transport chain, which I'm going to abbreviate with ETC, and then this is ATP synthase. Remember, ATP synthase is an enzyme making ATP, fusing together that third phosphate with adenosine diphosphate. It's our energy molecule. It's going to be used everywhere inside of our cell. So, what happens is we have NAD and FAD energy molecules, and what these proteins of our electron transport chain are going to do is they're going to break off those hydrogens. When they break off the hydrogen, they're going to release little electrons. I'm going to draw my little electrons as blue blobs here. These electrons are going to wiggle through the proteins of the electron transport chain. When they wiggle through the proteins of the electron transport chain, hydrogen is going to get sucked and pulled into that intermembrane space. So in my intermembrane space, I have lots and lots of hydrogen ions. And we've seen this before. We have seen a place where there are lots and lots of hydrogen ions. Hydrogen ions are going to flow from an area of high concentration in our intermembrane space to an area of low concentration in our matrix. Those hydrogen ions are going to get pulled through ATP synthase to make ATP. Remember the video we saw before, where where the hydrogen protons were flowing through ATP synthase, and ATP synthase was moving and turning to force together and mechanically make ATP. So, the whole point of glycolysis and the citric acid cycle is to get to this point of making NAD and fat energy. Because NAD and fat energy then give our electron transport chain the electrons that wiggle through, pulling hydrogen in to make the hydrogen gradient. This hydrogen gradient, hydrogen flowing through ATP synthase, is what gets us to that 30 to 40 ATP energy molecules. That's why I was telling you, NAD and fat energy are really the whole point of our citric acid cycle. Because those energy molecules run our electron transport chain, which allows us to get all of that ATP energy. One thing we haven't talked about is that electrons are dangerous. Electrons bouncing around inside of a mitochondrial organelle are going to cause some damage. Loose electrons, we have to grab them. We can't just let them bounce around. So when NAD and fat energy is broken, when we strip away the hydrogen, and those electrons wiggle through our electron transport chain, what's going to happen is we're going to have oxygen grab those little loose electrons. When oxygen grabs those loose electrons, it's immediately going to make water. Now, when our mitochondrion has made this water, we're going to have a high concentration of water in our matrix, lower concentration in our cytoplasm. Our water is going to go through osmosis. We could use the term hypotonic inside the matrix, hypertonic in the cytoplasm, lots of solute, lots of stuff. Water is going to move towards the solute. Water is going to move from a high concentration to low concentration. Water goes through osmosis to leave our mitochondrial organelle. So when we look at these metabolic pathways, we have to keep in mind that enzymes are proteins. So the shape is important. One enzyme is really only going to work on one substrate. They're either going to do a catabolic bond-breaking reaction or anabolic bond-making reaction. Water is going to go through osmosis. All molecules are going to go through simple diffusion. Bringing all of those concepts together, we have to keep in mind all the parts of the cell, what those parts do, what their function is, in order to understand this larger, truly amazing process. Because I'm not thinking about my cells right now doing glycolysis, doing the citric acid cycle, and making that hydrogen gradient after breaking NAD and fat energy, and running those hydrogen ions through ATP synthase. I'm thinking about how to talk to you about this and hoping that you understand this. I'm not thinking about all these details. My cells are just doing it, and I am grateful, because I don't want to keep all of this in mind every time I eat breakfast or dinner or have a snack, that we got to go through all these steps just to make ATP energy. So, going through this process and going through these steps is really going to give us a better understanding of our cells, tissues, organs, and the entire organism. And then we get to talk about what can go wrong. So, what if I don't have oxygen? If I don't have oxygen to catch these loose electrons from my electron transport chain, well, my cell could die. So, the amazing thing about most cells is that in that moment where oxygen isn't present, they can switch. They switch over to anaerobic cell respiration. They switch over to fermentation, which does not require oxygen. But in the case of anaerobic cell respiration, in the case of fermentation, none of this matters. Our mitochondrion cannot be used because our mitochondrion is depending upon oxygen for its internal processes. So, no oxygen, no 30 to 40 ATP. All you can do is glycolysis. Remember, glycolysis happens in the cytoplasm. We don't need oxygen. Our enzymes grab a hold of glucose. They transform it into pyruvate. In the process of doing that, they're going to make some NADH energy and a couple of ATP. So if you're a cell, you've had to switch over to fermentation, you've had to switch over to anaerobic cell respiration, then one of the problems is that you're only getting 2 ATP energy molecules. So this is a short-term solution because you know that you need hundreds and hundreds and thousands of energy molecules in order to keep your cell functioning and alive. But at least you don't die right away. Those 2 ATP energy molecules can keep you kind of limping along, can keep you kind of going. You're anticipating you're going to get oxygen again. If you don't, then your cell will die, your tissue will die, your organ will die, and the whole organism will die. So those are really the steps that lead up to dying from asphyxiation, dying from not having enough oxygen, is that each individual cell needs that oxygen. It can only survive for a short amount of time without oxygen. So we're really hoping we can get oxygen to those cells sooner rather than later. Now, one of the things that makes me really nervous and really worried about respiratory diseases is that if our lungs can't get enough oxygen, if we are not getting enough oxygen throughout our cells will start to flip over, right? Our cells in our kidney, our heart, our liver, they'll flip over and do fermentation, but they can't survive for very long. We get enough of those cells that die because they don't have enough ATP, then we see a tissue death. We get enough tissue death, the whole organ is damaged or potentially goes through catastrophic failure. And so keep this in mind. We've got a backup system, but this backup system doesn't last for very long. Now, normally when we talk about fermentation, we focus on pyruvate getting turned into alcohols, like what yeast will do. So yeast have this amazing ability. They're normally just single-celled little organisms. They don't necessarily need as many ATP as a liver cell or a lung cell or a heart cell. So yeast can take pyruvate and they'll transform it into alcohol, and they're going to be just fine. Of course, we like to imbibe alcohol, or we use it for things like hand sanitizer. But ultimately, if you can do aerobic, you want to do aerobic cellular respiration. If you have any questions, please, please, please send me an email.